Hi, my name is Dr. Katharine Hogan. I am the Director of Plant Conservation at Lauritzengardens in Omaha, and we're out in the field today to do some surveying and sample collections from one of our most imperiled species of plants here in the state of Nebraska, the yellow lady slipper, 𝐶𝑦𝑝𝑒R𝑑𝑖𝑢𝑚 parviflorum. It is a species that is pretty widespread across the eastern United States, but we're right on the western edge of its range, and it's estimated that there's not that many plants left in the state of Nebraska, so we're here to visit one of the largest remaining populations and see how many plants there are and do some sample collections for further conservation research. So Drew and Allison, they've both been out here multiple times, and they kind of think that the main clump is like a fairly small area, kind of in a circle, maybe like 20-25 meters radius. They're just gonna kind of do some kind of systematic walking back and forth. It's really hard to detect these guys in the midst of all the shrubs. Kind of walking back one way or another, kind of changing your perspective a few times gives you kind of a better chance at knowing that you've counted them all. One of the things we're worried about for plant conservation is how kind of resilient and stable the species is going to be over time. So at a population level, when we're in kind of one area where we know there's a bunch of plants, we need to know about how many there are, because if there's a lot of them, that's going to be a better indication that it's a really stable population, it's thriving, it's doing well. And if there's only a few of them, that could mean that they're either declining or they're experiencing some other kind of threat, whether it's drought or there's been a few of them for so long, they're kind of starting to get some genetic inbreeding. So basically we want to count to kind of get an idea of the overall health of the population. So when you see clumps like this, usually when a plant is young, it'll just put up a single stem, and then over time it starts gradually adding more stems. And these plants grow really slowly, so when we see one that's this big and has this many stems, it could easily be, what would you think, Drew, like a century old? Yeah. So orchids live in a pretty close symbiotic relationship. It's kind of a relationship of mutual benefit with fungi species that live underground, and they kind of colonize the roots of the orchid, and they exchange nutrients and energy, and they kind of both help each other survive. And we believe that those fungal partners are going to be key in hopefully restoring some of these endangered orchids, because they live in such close relationship with them. If we try to grow orchids in a greenhouse and then transplant them out without those fungi, they're probably not going to do very well, because they're missing those partners that they've evolved to grow with, essentially. What we're going to be doing is we're going to be taking a sharp soil knife, and we're just going to kind of delicately dig down right at the base of the plant and separate one root from the plant and snip it off. And we're going to send the root samples in these little baggies that have been sterilized off to partners at the Smithsonian Institution, who are going to try to isolate those fungi so we can determine who the orchids are growing with in their natural habitat. So we're kind of right on the western edge of the species range, so we're right on the western side of the Missouri River. And those kind of natural barriers, distance as well as physical breaks like rivers or lakes or bodies of water, can create what we call our disjunct populations, where there isn't really much genetic flow between the populations because the seeds don't disperse that far, or the pollinators don't have the range to kind of travel from one population to another. So it can kind of create these what we call disjunct populations that sometimes have really unique genetics from the rest of the species. As well as collecting the root samples, another thing we're going to be collecting while we're out here with these orchids is a piece of a leaf from a few individuals. And these leaves are also going to get sent to our partners at the Smithsonian, and they're going to have their DNA sequenced.
One thing that they talked about that was really important with collecting leaf samples for genetics is to get the plant tissue drying as quickly as possible to preserve as much of that for DNA extraction. So we put it into silica gel immediately after clipping to get it starting to dry out right away.
So one of the biggest things that people can do to help the conservation of this species is if you find the yellow lady slipper or any real orchid out in the wild, don't distribute the information, don't pick the plant, don't disturb it, try to leave its habitat as pristine and kind of untouched as possible. These are fairly sensitive plants and we need to be really careful how we interact with them. I think these plants are really special not only because they are beautiful and wonderfully aesthetic to look at and wonderful, but from a scientific perspective, some of these rare species have very unique roles within their communities. So they are able to support other pollinators, support pollinator species, and kind of fill different ecological roles that a lot of our common species can't. So they may be rare, but they're very important for the overall functioning and health of our native habitat.